This video will cover subtopic 6.1, Digestion and Absorption. Here are a list of skills, understanding, and applications that will be covered in this video. There are a few things to point out that you should be aware of as we go through this subtopic. So students should know that amylase, lipase, and endopeptidases are secreted by the pancreas, and the name trypsin and the method used to activate it are not required. Students should know that starch, glycogen, lipids, and nucleic acids are digested into monomers and that cellulose remains undigested. Tissue layers should include the longitudinal and circular muscles, mucosa, and epithelium, which refers to layers of the small intestine. So in digestion and absorption, it requires you to consider the structure of the digestive system. The various organs that make up the digestive system are all connected together and do perform specific functions to achieve digestion and absorption. And so we'll basically start at the top and work our way down. So we'll start with the mouth, and this is where mechanical digestion of food will begin. And this is done by chewing food using teeth, and this chewing action will also allow that food to mix with saliva. Saliva will contain mucus for lubrication, as well as amylase, for example, which will begin starch digestion. The next structure down is the pharynx. This is where the nasal passage and mouth meet at the back of the throat. This is where the swallowing process will begin here through the use of involuntary muscles. Now, because you have both the trachea and the esophagus and the trachea at the back of your throat, where that opening can lead to either tube, and remember, the trachea will lead to the lungs. The esophagus is what will lead to the stomach. So you want to make sure that food that you swallow ends up in the stomach. To ensure that that happens, there's a structure called the epiglottis that will cover the end of the trachea when swallowing, making sure that food goes down the right tube, essentially. Once food is successfully swallowed, it then travels into the esophagus, and this is where a food bolus now will move to the stomach through involuntary muscle contractions. Those involuntary muscle contractions are referred to peristalsis. And the esophagus will also secrete mucus for lubrication, so things move down smoothly. So after being swallowed, that food will go down the esophagus through a series of involuntary muscle contractions. The food just doesn't simply fall down the tube into the stomach. It is instead a series of muscle contractions that will guide the food and move that food bolus down the esophagus into the stomach. So in this process of peristalsis, this is what will begin in the esophagus and continues in the stomach and in the intestines. So peristalsis is an involuntary set of muscle contractions which is controlled by the nervous system. It involves contraction of the circular and longitudinal muscle layers that line the digestive system. Basically, that tube that runs from your mouth to the other end has two layers of muscle around it. So if this is part of our intestinal tract here, you have one layer of muscles which is referred to as the circular muscle layer. This is, if you're looking at it straight on, the muscles basically look like they're forming a ring around that tube. And then just outside of that, there's another layer of muscles referred to as the longitudinal layer of muscles. These muscles, or these smooth muscle cells, will run along the length of the tube, while the circular smooth muscle cells will run around the tube. So working together, these muscles create a series of contractions. So if this is our esophagus here, we have our food bolus moving in this direction down the tube. You'll get contractions behind the food bolus that will squeeze the tube inwards, forcing that food bolus down the tube. And it is a series of contractions that will allow that food bolus to move down the esophagus. Now, there are several things that peristalsis does that are good. So peristalsis only will go in one direction, and this is what allows you to eat upside down. Or if you don't eat upside down, well, you've probably eaten some potato chips or something while lying on the couch or in bed. And when you swallow those potato chips, that's what allows that food to then move from your esophagus into your stomach. Now you can have antiperistalsis, which occurs, which is known as vomiting, and this involves the skeletal muscles of the abdomen and not the smooth muscles. Basically, those are the skeletal muscles in your abdomen, kind of causing contractions in your stomach, forcing that food out the other way. Peristalsis will also serve to churn the food and will allow it to mix with enzymes for digestion. So now from the stomach, that food bolus will now enter the stomach from the esophagus, and in the stomach, this is where that food bolus will now churn and mix with water and mucus and stomach acid. So the stomach will start a series of contractions that will mix the food around with water and mucus and stomach acid that's found in the stomach. Now that stomach acid, which is basically hydrochloric acid at a very low pH, about a pH of 2, will work to kill bacteria and other pathogens as well. Because you have to remember that the intestinal tract is basically a tube open to the outside environment, and as that food bolus now will start to work through your system, you want to try and keep bacteria, unwanted bacteria and other pathogens out, so that stomach acid works as a layer of protection. Protein digestion will also begin in the stomach through enzymes referred to as endopeptidases. They will start to cleave peptide bonds and start to break down large proteins into polypeptide chains or shorter protein fragments. Because there is stomach acid found, because there's that acid found in the stomach, and there are cells that are lining the stomach, there is a layer of mucus that will protect the lining of the stomach from that acid, and will prevent that acid from damaging those cells. From the stomach, that food bolus will then enter into the small intestine, and it is here where the final stages of digestion will occur in the upper part of the small intestine by enzymes. The stomach acid that is accompanying that food bolus is going to be neutralized by bicarbonate secreted by the pancreas. This will also increase the pH to be slightly alkaline. And it is in the small intestine where nutrients get absorbed. So the final stages of digestion, where those molecules get broken down into individual monomers, those nutrient monomers then get absorbed by finger-like projections in the intestine known as villi. So there are these finger-like projections that point towards the lumen or the opening of the small intestine, and it is on these finger-like projections where absorption will occur. From the small intestine, that food bolus will then travel into the large intestine, or the colon as it's sometimes known, and then the large intestine will function to absorb water. In fact, a lot of water absorption occurs in the large intestine. There's a lot of water associated with digestion, and then your body works to recover a large portion of that water. This is also where you get formation, storage, and eventually egestion of feces, and then absorption of minerals will occur in the large intestine. There is minimal nutrient absorption, and the large intestine will basically end with the rectum and then the anus. One of the skills you'd be expected to do is to be able to identify tissue layers in transverse sections of the small intestine, viewed with a microscope or in a micrograph. So you should be able to identify the serosal layer, the circular and the longitudinal muscle layers, and the submucosa and mucosa. So we'll go through a few pictures where you'd be able to identify these various layers. So the picture here is just a diagram of these different layers. So this is the small intestine. Here we have the lumen, which is the white space. This is basically the opening. This is where food would travel down. You have the villi lining the small intestine. These are those finger-like projections. And then outside of the villi is going to be the circular smooth muscle layer, and then the longitudinal smooth muscle layer outside of that. So in a micrograph, what would that look like? So if we have a look at our picture here, the white space next to the purple is going to be the lumen, and then the purple area here is going to be the villi, and then just below that purple area where we get to this kind of reddish pink area, this is going to be our smooth circular smooth muscle layer, and then this kind of lighter purpley area is going to be the longitudinal smooth muscle layer. Looking at a different transverse picture, we can still identify the same layers. So here in this white area, we have our lumen, and then this purpley area where we have these finger-like projections. This is going to be our mucosa, where we'll find the villi. And then just below that purpley area is going to be the submucosa. This is going to be composed of connective tissue, blood vessels, and some glands. And then just below that we have the muscularis layer, where we can find our circular smooth muscle layer and our longitudinal smooth muscle layer. And then at the very bottom we have our serosa, which is basically like connective tissue, just kind of holding everything together. So in this process of digestion, most food molecules, remember, are large polymers and are going to be relatively insoluble. So in order to be absorbed into the body, they must first be digested into smaller soluble molecules before they can be absorbed into the blood. So this diagram just runs through what that process of digestion would look like for our different macromolecules, for carbohydrates, lipids, and proteins. So for carbohydrates, and the main carbohydrate that we might ingest would be starch. That starch is going to be broken down by the enzyme amylase into the disaccharide maltose. And then from there, there's a separate enzyme, maltase, that will break down that maltose into glucose. So maltose is made up of two glucose monomers. Maltase will break apart that disaccharide into glucose units. Lipids, which will be digested by lipases, first have to be emulsified, and this is basically a churning action of the stomach. And by bile salts secreted in the duodenum. Basically, those bile salts are going to come from your pancreas, and they'll help to make those lipids more accessible to enzymes. So once they've been emulsified, you'll have the enzyme lipase, which will break down those fats into fatty acids and glycerol components, so into those individual units before they can be absorbed. Proteins will have to first be digested by endopeptidases. Those are found in the stomach, and they'll be broken up into shorter polypeptide chains. And then you'll have a series of different peptidases that will break down those shorter polypeptide chains into dipeptides, so 2 amino acid units. And then you have dipeptidases that will take those dipeptides and break them down into those individual amino acids. And then it is those individual amino acids that will get absorbed into the body. So there are additional organs that will contribute to this process of digestion, but not be directly involved. So those organs are the pancreas and the liver. So both will secrete digestive enzymes into the lumen of the small intestine. The liver will produce bile, which is stored in the gallbladder, and then released into the large intestine. That bile will be used to emulsify fats. Pancreatic juice will contain enzymes that digest all three macromolecules, those fats, carbohydrates, and proteins. So you'll have amylase, as I mentioned in the previous slide, that will digest starch, lipase that will digest triglycerides, and phospholipids and proteases that will digest proteins. So there are a number of different enzymes that are secreted by the pancreas, which can include amylase, lipase, phospholipase, and proteases. But then there are other glands in the intestines that will produce more enzymes for further digestion. So these enzymes will break down those products into smaller fragments, and then you have other enzymes that will break down these molecules further, for example. So you'll have maltase that will break down the maltose into glucose. You'll have exopeptidases that will break down shorter peptides into dipeptides and amino acids. You can have nucleases that will break down DNA and RNA into nucleotides before they get absorbed. And then just keep in mind that cellulose, which is the main component of the cell wall in plants, is not digested. So it's not broken down, as humans do not have the enzymes to break those beta 1,4 bonds found in cellulose. It continues through the digestive tract and basically is excreted as waste. However, it is important as it provides the diet with fiber. So cellulose, which is found in plant cell walls, is also known as fiber. So eating fiber is important for maintaining a healthy digestive tract. In starch digestion, as mentioned, starch is too large to pass through the villi in the intestine. So just recall there are two main forms of starch. There's amylose and amylopectin. Both are composed of those alpha 1,4 glucosidic bonds. However, amylopectin has alpha 1,6 bonds at those branch points. So amylopectin is going to be branched, amylose is going to be linear or helical. The digestion of starch is exothermic and will begin in the mouth with that enzyme amylase. Salivary amylase stops working when it reaches the stomach due to the acidity. The enzyme becomes denatured and can't work anymore. In the small intestine, starch digestion will continue by pancreatic amylase. So the pancreas will produce amylase as well, which will continue that starch digestion. Amylase will break down the 1,4 bonds in starch into 2 and 3 glucose fragments called maltose and maltotriose. The 1,6 bonds of amylopectin cannot be broken down by amylase and are called dextrin. Now, the digestion of maltose, maltotriose, and dextrins will be completed by the enzymes in the membranes of the small intestine, and those enzymes are maltase for maltose, glucosidase for maltotriose, and dextrinase for dextrins, and these will all break down these smaller fragments into glucose monomers. Once glucose has been produced, glucose is then absorbed into the villus by co-transport with sodium ions. That glucose then enters into the capillaries in the villus, and then the venules into the submucosa of the wall of the small intestine. The blood in the venules is then carried by the hepatic vein to the liver. So basically, once that glucose has been absorbed into the villus, it enters into capillaries, which are then carried off to the liver through the hepatic vein. And then once in the liver will store excess glucose in the form of glycogen for energy storage at a later point. So we'll quickly go through the role of the villus in absorption of the end products of digestion. So with the structure of the villus, basically the villi are structured so that it will increase surface area for maximum absorption of end products of digestion. So if this is our small intestinal tract, you'll get these folds on the intestinal wall, which will help increase the surface area. On those folds are basically these finger-like projections called villi, which are helping to increase the surface area over which absorption can occur. And then on those finger-like projections is where you'll find individual cells. So those finger-like projections are lined with cells called epithelial cells. And on the surface of those epithelial cells, they have folds on their membrane called microvilli. And it is on the microvilli that you find the enzymes for the final stages of digestion, and where nutrients will then get absorbed into the body. So those microvilli will help increase the surface area that is available. So this image here shows the structural adaptations of the villus as well. So here you have the epithelial layer where nutrients get absorbed. So we have the lumen where the food will be found, or the nutrients will be found. From the lumen, nutrients will be absorbed from the epithelial cells into those blood vessels, which will then be transported away to the liver. And then the green structure is referred to as the lacteal, or these are the lacteals that will be involved in lipoprotein transportation. So those fats that get absorbed into the body come in through the lacteal. So in absorption by the villi, that epithelium that covers the villi will form a barrier to harmful substances. So those epithelial cells will create a barrier that will keep harmful things out and only allow good things through. So only small monomers, vitamins, and water can essentially pass through. So the products of digestion of those macromolecules that get absorbed are things such as monosaccharides, such as glucose, fructose, and galactose, amino acids, fatty acids, such as monoglycerides and glycerol, and nucleotide bases, the A, the T, the C, the G, and the U, all of which get absorbed individually into the body. So there are different ways in which materials get transported into the small intestine. So there is what's called co-transport, which is a form of active transport. This is how glucose gets into the body, for example. So if this is our lumen, and then here we have a glucose molecule. Glucose molecule will be brought into that epithelial cell and ultimately through the body by co-transporting it with sodium that's found in the lumen of the small intestine. There is also facilitated diffusion. This is for other types of monosaccharides where there'll be a channel that will allow that monosaccharide to move from high concentration to low concentration, and the cell will work to keep that monosaccharide in low concentration inside the cell as well by moving it quickly outside. Water will come in through osmosis, and then triglycerides will move in through simple diffusion. So this picture here just goes through the various stages of food processing that we've all talked about, just kind of keeping in mind the bigger picture. So we have digestion, or ingestion, sorry, which is that process of taking food into the body. This is where the process of digestion will then occur. So this is breaking down that food through the process of mechanical digestion or chewing food, and then you have chemical digestion occurring where enzymes break down those polymers into monomers. And then once digestion has completed, then the process of absorption can begin. This is where food will then be moved into the bloodstream and then transported to the necessary cells that will need it. Once those nutrients are in the cells, this is where they get assimilated, making that food part of their own cell. So amino acids that have been taken up from the food that has been eaten is used to make proteins that the cell would make. nucleotide bases that have been taken up from or absorbed through the food are then used to make things like RNA for the cell or used in DNA replication. And then the final stage is elimination, and that removal of any undigested materials. So one of the activities that we will perform at some point will be modeling the small intestine, so you can model that movement of smaller substances through the intestinal wall through something such as dialysis tubing. Dialysis tubing will have small holes in it that will only allow for the movement of small molecules through it. If you place starch in the middle or in that dialysis tube, tie off the ends, and then place an enzyme such as amylase in the tube, you can determine how those molecules are moving. So starch will not move out of the dialysis tube, but small molecules such as glucose will, and as maltose will. And as maltose is formed from starch digestion, it can move out of the tube, and you can test for the presence of maltose outside of the tube in the beaker water or inside the dialysis tube as well. And you can test for the presence of starch inside of the dialysis tube as well as outside the dialysis tube. More information on this can be found on page 287 of your textbook. And the last part that we will do is we will go through producing an annotated diagram of the digestive system, which we will cover in class. So that is the end of this video for topic 6.1 on digestion and absorption.